Today I'm going to be showing you guys how to remove all of this non-specific background staining that we've got here. First we're going to go to Image, Color, Split Channels. Now that's just going to take our channels and separate them so that we can edit them separately. They might keep their color or they might turn black and white like they did here. Either way, it doesn't affect anything that we're going to do next. Now we're going to select one of our channels. I like to start with my Dappy, my blue. We're going to go to Image, Adjust, and Brightness Contrast. Now I'm trying to get rid of all of this gray here. I'd really like it to be more so black and white instead of having this mid-tone. So we're just going to start messing around. I really want the space in between my cells, especially with Dappy, to be almost completely black. One thing that I have to remember though is that Dappy is really, really bright, especially compared to some of the weaker antibody stains. And so I've got to make sure that I'm toning down the brightness a little bit more than I would with my other channels. All right, so that looks good to me. I'm just going to go down to apply. And then this little window is just telling me that it's going to overwrite the image information that it started with. So we're going to make sure that we already had it saved in just completely raw somewhere else, just in case we need to start over, because this is going to actually permanently edit the pixel values. Great. Now we're going to our next one. This is our red channel. We're going to go same thing, adjust, brightness, contrast. Now, especially with our non-Dappy channels, we want to start with bringing up the minimum when we've got a lot of background. That's just a really good place to start. And then we can start messing around with the brightness and the contrast. I'd like this to be brighter than it is, just by a bit. And now I've got to up the contrast to the point where I no longer see some of that non-specific staining where all those cell bodies were. Because I know that my stain is not supposed to be in that area. And I don't really want that color anywhere that the antibody is not supposed to be. The best I can anyway. So I like that. We're going to go to apply, same as before, and fully apply it. Now, this is the last one. This is our green channel. Not a great stain. We've got a lot of background, and the values that are really well stained aren't really that bright. Here we're going to have to mess around a little bit. We might have to up the brightness and contrast a lot more than we did our other images. We'd really like the maximum values of each channel to kind of match, which means that we're going to have to probably tone down Dappy like we did before, but some of these weak stains we've got to really tone them up. So now we finally got rid of all that kind of non-specific background gray color. So we're going to hit apply, fully apply it, and now we're ready to see what it looks like all together. So we're going to go to Image, Color, Merge Channels. Now we have to make sure that all of the channels are in the right spot. So these ones are really nicely labeled for ImageJ by us, so it has the parentheses in red so that you know it goes to red. But that's not always the case. And so sometimes it's going to call it C1, C2, C3, but it might not remember exactly what color it is. And so we have to make sure that we look at every single one in those cases to make sure that the red is actually the red and the green is actually the green. So we've got a pretty decent image here. We can see a nice colocalization in that yellow color between the red and the green. We see that the Dappy isn't so overblown that it's overtaking all our other stains. We can see that we still kind of have a little bit of that non-specific green stain, but with such a weak stain, we're probably not going to get much better than this. So especially if we're comparing to what we started with, we have a lot less background this time, and it's a little bit of an easier image to look at. Alright, so as a little addendum, I kind of wanted to take you guys through the ImageJ wiki. ImageJ or FIJI is an open source software, which just means that anybody can edit how it functions and create little plugins for it that allow it to do new things. So we're not waiting on a development team to do all of this, we can kind of do it ourselves. Which means that ImageJ has a lot of functionality that some of even the best softwares out there don't really have. So I just wanted to show you that if you wanted to learn new things, or if you ever needed help with ImageJ, how it works, why it's doing what it's doing, you can always talk to the TAs, but it might be quicker, especially if we're in the lab late at night, to be using this ImageJ wiki. So we can go tutorials, user guides, tips and tricks, troubleshooting, frequently asked questions, and we can even extend into plugins. So these plugins are extra bits of software that can help ImageJ do things that it normally wouldn't be able to do. I actually, for my project, use Standard Neurite Tracer, which is a plugin that allows me to do what's called shoal analysis, which is a tracing of the different branching patterns of neurons. And then we can actually take a look at them and figure out where exactly they are being branched as far as distance from the center goes, or the nucleus. And we can kind of see exactly what morphological changes might be a little bit more difficult to describe otherwise, especially quantitatively. And I only learned how to do that through the wiki, um, maybe a couple of YouTube videos, but this is a really, really great resource that you would be really smart to properly utilize.